Okay. So, fatty acid biosynthesis. The de novo pathway of fatty acids involves three stages. First is the translocation of acetyl-CoA from the mitochondria into the cytosol with the help of malate shuttle. This is followed by the carboxylation of acetyl-CoA with the help of acetyl-CoA carboxylase, which is a multi-enzyme complex. And the carboxylation is carried out in presence of biotin because carbon dioxide is donated by the bicarbonate ions. This is followed by the conversion of the acetyl-CoA to its next higher member, that is a malonyl-CoA, and this malonyl-CoA can now enter into the de novo pathway, and this forms a committed step of the reaction, and the fatty acid biosynthesis is initiated with malonyl-CoA in one arm and acetyl-CoA in the other arm. And with this malonyl-CoA, it is now responsible for carrying out or initiating the de novo pathway, and the de novo pathway simply involves the reversal of, almost the reversal, of course the enzymes can vary, but the reversal of the beta oxidation in which the two carbon moieties, acetyl-CoA molecules in the form of malonyl-CoA condenses together to finally give rise to the C16 palmitic acid in case of all the tissues except the lactating mammary glands. In the case of lactating mammary glands, the fatty acid will be a short chain one. It will not be elongated to a long chain fatty acid. Right. So this is what is happening, and the malonyl-CoA, conversion of malonyl-CoA into a long chain fatty acid. Actually, it is not the malonyl-CoA. Malonyl-CoA immediately undergoes decarboxylation. So the fatty acid synthesis have carried out with the help of acetyl-CoA molecule itself. But what is the role of malonyl-CoA? Because the conversion or the condensation between two acetyl-CoA molecules is not possible because it's a thermodynamically unfavorable reaction. It's a high energy requiring process. So the acetyl-CoA has found an alternative, that is acetyl-CoA that is entering into the de novo pathway will be first undergoing the carboxylation reaction to get converted into malonyl-CoA. So only a single molecule of acetyl-CoA will be entering into the fatty acid synthesis in a single step. All the other acetyl-CoA molecules will be entering in the form of malonyl-CoA, and once the fatty acid biosynthesis initiated, it immediately undergoes decarboxylation to give rise to acetyl-CoA itself. So net effect is that the two carbon acetyl-CoA molecules are condensing together to give rise to the long chain palmitic acid, except for the acetyl-CoA is not coming in the form of acetyl-CoA. It is coming in the form of malonyl-CoA, except for the first molecule. Only one molecule, a single molecule of acetyl-CoA will be entering in the form of acetyl-CoA itself. All the remaining, for example, if C16 palmitic acid is being synthesized, 8 molecules of acetyl-CoA are required. And out of this 8 acetyl-CoA, only single acetyl-CoA will be coming in the form of acetyl-CoA itself. All the remaining acetyl-CoA molecules are entering into the de novo synthesis in the form of malonyl-CoA by undergoing the carboxylation reaction carried out by acetyl-CoA carboxylase. Clear. So once this malonyl-CoA is entered, is initiating the de novo pathway, the further reactions are carried out by another multi-enzyme complex which is known as the fatty acid synthase complex. So what's the structure of, so both the acetyl-CoA carboxylase and also the fatty acid synthase, FAS, we can cut short, we can abbreviate the fatty acid synthesis FAS. So FAS or fatty acid synthase complex is also a multi-enzyme complex like that of acetyl-CoA carboxylase. And this fatty acid synthase complex, its structure itself is very complicated because it's a dimeric molecule. That is, two monomers are sitting side by side in a head-to-tail fashion. That is, the head of one monomer is facing the tail of the other monomer and vice versa. Clear, the tail of the first monomer will be facing the head of the second monomer. So two molecules, two monomers are arranged in an adjacent manner, in a side-by-side fashion, in a head-to-tail fashion again, to carry out the synthesis of fatty acids. Clear. So the multi-enzyme complex, the fatty acid synthase complex, consists of 7 autonomous enzyme units. We can name them as E1 to E7. In case of PDH complex or alpha keto glutarate dehydrogenase, etc., we name them subunits as E1, E2, and E3. Similarly, in case of fatty acid synthase complex, we can name the enzyme, individual enzymes, as E1 to E7 for simplicity. Of course, the actual name is something different, but we can simply now, for the time being, we can designate them as E1 to E7, right? And there are 7 monomers in a single subunit of fatty acids. So a set of two sets of 7 enzyme units are coming together in a head-to-tail fashion to carry out the synthesis of the fatty acid. And we can again, since two sets of E1 to E7 are there at a time, or once each round of fatty acid synthesis involves the formation of acetyl-CoA molecules, 2 molecules of acetyl-CoA at a time, because both are functioning in an independent manner also. Again, they are working in a orchestrated fashion, but they are individual units. Clear. So at a time you can have two molecules, or that is 2 sets of acetyl-CoA molecules will be undergoing condensation at a time, right? So this is the acetyl-CoA carboxylase. It's a carboxylating enzyme responsible for converting acetyl-CoA to malonyl-CoA. This is a committed step of the reaction, because if malonyl-CoA is formed in the body, it is solely channeled into or funneled into de novo pathway, right? And this enzyme requires biotin as a coenzyme. Biotin is actually undergoing the carboxylation reaction, or the carbon dioxide is carried from the bicarbonate ions towards acetyl-CoA by the biotin. So biotin first undergoes carboxylation by biotin carboxylase, and this carboxyl group is carried from the biotin towards the acetyl-CoA by biotin carboxyl carrier protein, and the trans carboxylation reaction happens. So all these are enzyme activities functioning in acetyl-CoA carboxylase. So that's why it is known as a multi-enzyme complex, because it is made up of 3 enzyme units. That is, it's carrying out first the biotin is undergoing carboxylation by biotin carboxylase enzyme, converting biotin to biotin carboxyl carrier. So, or carboxybiotin. Biotin undergoes carboxylation to give carboxybiotin. This carboxybiotin again is carried by another protein enzyme known as biotin carboxyl carrier protein, taking the carboxyl biotin towards the acetyl-CoA. And what does this carrier protein do? It transfers this carboxyl group from the biotin, carboxybiotin, to acetyl-CoA, converting it into malonyl-CoA and converting the carboxybiotin back to biotin. So all these are 3 enzyme activities are carried out in the same as a single molecule of acetyl-CoA. Hence, it's a multi-enzyme complex. Again, it's also having an allosteric site to which the bicarbonate comes and binds. Clear? So this forms the structure of the acetyl-CoA carboxylase enzyme. So the regulatory or allosteric site to which the activator or the inhibitor come and bind. Right? So it's a very important enzyme involved in the fatty acid synthesis. Now, what about the fatty acid? Now we are having our malonyl-CoA with us. Now this malonyl-CoA is entering into the de novo pathway, and the de novo pathway involves the biosynthesis of the fatty acid. Clear? And each round, the beta oxidation is 4 set and another oxidation, hydroxylation, again oxidation, then thiolytic cleavage. Here in the case of synthetic pathway also 4 steps. Each round of each round of fatty acid synthase complex reaction involves 4 reactions, right? One round of condensation reaction. 2 acetyl-CoA molecules condense what will happen? It gives rise to 4 molecules of fatty acid which is having 4 carbon chains, right? Oro pravisham cycle end the chain will be lengthened by 2, right? The beta oxidation the chain was shortened by 2. Here the exact reverse happens. But 2 plus 2 4. So when 2 molecules of acetyl-CoA condenses together, it gives rise to butyl-CoA. Butyl-CoA again enters into next round of fatty acid synthesis to give rise to 6 carbon fatty acid. 6 carbon is converted into 8 carbon. So in each round, when each round of fatty acid synthesis ends, the resulting product will be having a chain length increased by 2 carbon. Okay. And the reaction is carried out by a multi-enzyme complex. So only after all the reactions ends up, then only the product will be released, right? As such in the case of our PDAs on, right? So the fatty acid synthase complex, it's a polypeptide protein. It's a protein having multi-enzyme activities, and there are 7 enzyme activities. PDS like a moon and acetyl-CoA carboxylase la moon and but in case of fatty acid synthase complex is a very complicated structure because it's having 7 different autonomous items. Oro enzyme independent activities and so autonomous 7 autonomous enzyme activities, and which can be named as E1 to E7, and it is present in yeast, mammals, lower organs, right, and can carry out the fatty acid biosynthesis. And it's a dimeric molecule, that is two set of E1 to E7 in there, and they are arranged in a head to tail fashion. This is if suppose this is E1 to E7 elliptical model fatty acid synthesis. So if this is one set of E1 to E7, the other set will be facing in the inverted manner. That is the head. Suppose if it is the head of the monomer 1, the head of monomer 1 will be in contact with the tail of the second monomer. Clear? Second monomer, it will be getting contacted like this. So in a head to tail fashion. So an elliptical shape. This fatty acid synthase elliptical shape. Clear? Like this. It's like a ellipsoid. So only after the 4th reaction is over, then only the product will be released out. Again, a product then the first enzyme find it and the pathway will be continued. Right. So the use of the multi-enzyme, again, multi-enzyme, so that after each reaction, the product is not allowed to come out. Right. So there is a compartmentalization happening. So synthesis of all the enzymes is coordinated since it and all the 7 enzymes are coded by a single gene. So what is fatty acid synthase? It's a polypeptide chain having 7 enzyme activity, E1 to E7, and in addition to this E1 to E7, there's one more thing which is known as acyl carrier protein. Structure I'll show you. It's a long molecule. Now, oro enzyme activity enzyme biotin. TPP in case of PDS, it was a TPP which is acting like a swing. In the case of fatty acid synthase complex, the molecule which is acting as a swing to carry one molecule from one location to the other. Even the product in E2 E3 it is acting like a swing. A swing molecule is there, which takes the product from one side to another enzyme. And that swing is the acyl carrier protein. So E1 to E7 are there. In addition to it, it is having an acyl carrier protein. Now, which are the enzymes? That is acetyl transacylase, malonyl transacylase, then 3-keto-acyl synthase. The short forms are AT means acyl transacylase. Acetyl transacylase. This is MT means malonyl transacylase. This 3-keto-acyl synthase. Then 3-keto-acyl reductase KR. Then enoyl reductase ER. Then there is a thioesterase. After which what comes? ACP. Clear? I'll show you. In ACP swing polling free at end. We call it as PAN-SH, which is made up of pantothenic acid. So pantothenic acid, it is in the form of 4-phosphopantothene, SH group moiety. So this moiety SH group travel swing move clear. So it is having a pan-SH and a cis-SH. I'll show you the structure of fatty acid synthase complex. So this is the structure of, this is a dimeric form of fatty acid synthase. You can see the partition. So this is monomer 1. The acetyl transacylase, malonyl transacylase, keto-acyl synthase. Then this hydratase, enoyl reductase, keto-acyl reductase, and finally ACP. This is the 7th protein. This is the molecule which is acting as a swing. And the thioesterase activity now finally. This enzyme comes only at the end. Last round otherwise, so this is the 7 units of monomer 1. Monomer 1 suppose this is a head. Head and then the cis-SH moiety. Cis means cysteine. Cysteine is a SH group. This is facing ഇതാണ് ഇവിടുത്തെ മോണോമർ 2. ദിസ് ഇസ് മോണോമർ 2. ഇത് മോണോമർ 1. ദിസ് ഇസ് മോണോമർ 2. മോണോമർ 2ന്റെ ACP is here. So ACP ക്ക് എന്താണ് 4-phosphocandethane. അതിനും ഒരു SH ഉണ്ട്. Clear? So cis SH and pan SH are facing towards. So ഇതാണ് ഇത് ഇതിന്റെ ഹെഡ്, ഇത് ഇതിന്റെ ടെയിൽ. ഇത് മോണോമർ 1ന്റെ ടെയിൽ, ഇത് മോണോമർ 1ന്റെ ഹെഡ്. So both are acting in a head like fashion. Head